Good morning, everyone. So, as it was mentioned, I'm from ProDigest. I'm a co-founder of the company, and we are expert in simulation of the gastrointestinal tract. So we develop models to recreate on a bench what is happening along the digestive steps in our gastrointestinal tract. Our mission is actually to help clients to de-risk their product development, and we are able to do that because by using our technologies, we provide mechanistic information of how a given ingredient, a given active is working in the gut, and in this way, actually, we help them to decide on the best leads to move on then further with the research. And essentially, all our technologies are in fact validated with in vitro and in vivo data. Now, which are the main pillars, the main drivers currently in the market? First of all, you are well aware of the 3R principle: reduce, refine, and replace animals in research. So when you want to basically apply this principle, you must use technologies that, of course, are able to recreate the complexity of the gut in a proper way. So you have to be able to look into the different micro-niches that are characteristics of the gastrointestinal tract. For instance, lumen versus mucus, or upper gastrointestinal tract versus different areas of the colon. Second big driver is about personalization. Nowadays, it's all about precision nutrition, precision medicine. Hence, you must take these factors into account. And therefore, when you work with the technology, this technology must be able to reproduce the physiology of the specific target population. And within a target population, being a healthy population or a diseased population, multiple individuals, because each one of us has a different microbiota, and such variability can play a role on the outcome of the study. Last but not least, throughput versus detailed mechanism of action and complexity of the simulation that we perform. We do strongly believe that it doesn't exist the single technology that can answer all the questions. In fact, depending on the research question, you have to select the right technology. With this in mind, at ProDigest, we develop multiple technologies that actually can be used to, in fact, assess what is happening in the gastrointestinal tract. When you look at the slide, when you go on the left side, you have the colon on a plate, which is in fact our technology with the biggest throughput. You can really look into mini reactors of 7 ml and test hundreds of conditions in parallel to have a very high throughput. And the more you move from left to right, you increase the complexity of the simulation to end up on the right part with our SHIME, which is our flagship technologies, which is a simulator of the human intestinal microbial ecosystem. And essentially, it's a computer-controlled device with many reactors, each reactor representing a part of the gastrointestinal tract, so stomach, small intestine, and different areas of the colon. All systems, of course, we talk about the personalization, has been adapted and validated to be representative for different human conditions, different age, healthy condition, disease conditions, but also we have systems to look into animals, mainly pet industry and pig. Now, how to select the right system? We have clients coming from all over the world saying like, well, I want to do a SHIME study. Then by brainstorming, we do realize that maybe the SHIME is not the right fit. Or the other way around, I want to do a colon on a plate, maybe the SHIME is a better fit. So the idea here is just to give you like 3 examples to see how basically for a specific research question, how you would select a specific tool to do the research itself. First work was a collaboration with the Les Affres, in which we have been asked to test 4 different types of breads, sourdough or regular yeast bread, and each one with a white flour or a whole grain flour. And the aim was to assess the digestibility of the bread, and then, of course, to see the impact of the bread at the level of the microbiota in the colon, taking into account the intra-individual variability. So we are talking about a study with a high number of conditions, hence the need possibly of something with a throughput approach. So basically what we have first digestibility, upper gastrointestinal tract, and then the throughput approach with the colon on a plate. Essentially, when we look at the experimental design, we had the 4 breads. Each bread underwent the digestion steps, so first a mouth simulation, then simulation of the small intestine, use of a dialysis to look at the removal of what normally is being digested, and then the undigested fraction enters the colon on a plate, in which we are using 8 different donors to look into the intra-individual variability. So overall, we are talking about 40 conditions, 40 arms in parallel to be tested. So as I said, need of the throughput. Using this type of approach, actually, we were able to look at first bread digestibility. All bread show very good digestibility in terms of mineral release and absorption, in terms of carbohydrates, in terms of proteins. There were no strong differences between the different type of bread. At colonic level, we have seen a significant increase with all the different breads in terms of short-chain fatty acid production and a decrease in branch short-chain fatty acid production, which are linked to protein fermentation. There were no differences between the bread, mainly an increase in acetate and then propionate and butyrate among all the different breads. And then when we look at the microbial community, we have seen the increasing concentration of specific species, which are health-related species, such as, for instance, Faecalibacterium plasnitzii, butyrate producer, Roseburia, or Bifidobacteria. But overall, it was interesting to look, when we perform a principal component analysis, that at the very end, the inter-individual variability still played an important role. The variability of the host was still the main factor for which the samples were clustering together. So, using this approach, we're actually able to look into the digestibility of the bread and then the impact on multiple donors. In another research question, it was a collaboration with Nutrilits and together with the University of Wageningen, we were asked to look into the impact of benicaros, which is a fiber derived from carrot rhamnogalacturon, and to look into the structure-activity relationship and the gut microbial community. Here, the questions were completely different. They were looking into the structural changes during the fermentation step. They were looking into the impact of multiple donors and how this could have an effect on the structural changes. And then the possible impact of multiple daily doses when actually consume the fiber for several days in a row. So this is a research question that has a temporal factor in it, the multiple repeated dosages, and also a spatial potential effect. Hence, as you can imagine, the column on a plate is not a fit in this specific case for the question. Here we were actually looking more into an application of our long-term simulation with the SHIME, in which basically we are going to recreate a clinical trial in vitro for several weeks in a row. So this was the experimental design. So the test products were actually added in the SHIME, and this was done every day, breakfast, lunch, and dinner, like you have with a regular person after a control period, a treatment period. And then we look into the fate of the test compounds in terms of degradation and digestion of the compound, fermentation of the compound, and then add the impact on the microbial community, activity, and composition. We found something very interesting. So first of all, we found out that despite the complex structure of the compound, the product was mainly fermented in the proximal part of the column. So mainly fermentation was happening proximally where you have a strong saccharolytic activity. Then we identified two different donor-dependent fermentation pathways. When the donors had a microbial community enriched in Bacteroidetes, basically what was happening, you had a general degradation. So all the different components of the test products were all degraded at once, at the same time. On the contrary, when the donors were enriched in Prevotella, you have actually a preferential degradation. So basically you were starting to attack the compound from the external side and then continue the degradation internally. So two different pathways of degradation depending on the microbial community, the release of oligosaccharides and then the fermentation by these oligosaccharides by bifidobacteria in the arabine side chains, and then an increase in the concentration of bifidobacteria. So overall, two ways of consuming the product depending on the compound, on the microbial community. But the interesting factor was that over 3 weeks of time, basically both type of pathway led to the same type of metabolic outcome, meaning that the products were highly consumed and almost completely consumed during the fermentation period, and at the same time were leading to the same type of short-chain fatty acid production independently by the background microbiota. Third example is an example in which we actually were asked to work with a very low dosage of a plant extract and to look at the impact on gut wall modulation. So what actually was happening on gut barrier activity and production of pro-anti-inflammatory cytokines. And also we wanted to take into account the microbial variability, of course. Here we wanted to answer to a specific question. Is a single dosage good enough to see an activity, or maybe daily repeated dosages could actually offer a better outcome? So we wanted to compare two different technologies and see the main outcome of that. Selected technologies have been, on one side, the colony plate. Second case, the screening shime. Screening shime, I didn't mention it yet, but in fact is a different use of the shime itself. Essentially, each reactor in the configuration represents a colony compartment. And so basically you have multiple donors in each reactor, and actually you are able to look at the effect of daily repeated dosages over a period of 8 days. So here the experimental design is very simple. Plant extract, either we use 10 donors, so either colony plate or screening shime, and then the lumen, so the sample that we collected from the different reactors, were exposed on a co-culture containing CACO2 and THP1 cells. So it's a simulation of a leaky gut condition, and then you can observe whether the extract plus all the metabolites produced by the bacteria can exert a positive activity at the level of the gut wall. Now, what we found are the results. On the top part, you have the results obtained on TER, so transelatical epithelial resistance, which is an indication of the gut barrier activity, and IL-10, which is a pro-inflammatory cytokines, anti-inflammatory cytokines. Top part is the colony plate, lower part is the screening shime. As you can see, when we look at the results and the impact at the level of the gut wall using the samples from the colony plate, we have seen no differences between the control and the treatment, so no difference between blue and red bars. On the contrary, when we look at the lower part, we could see that the plant extract was able to induce a positive increase in TR, so strong gut barrier activity, and a positive improvement in IL-10 production. So from a mechanistic point of view, why this is happening? Well, if you look at a very simple parameter like butyrate, which is actually a compound known to have an anti-inflammatory activity, you could see that in the colony plate, there were no differences in butyrate production at the level of the colony plate. On the contrary, with daily repeated dosages, you could actually induce a good cross-feeding, and by the end of the 8 days of treatment, you were able to have a proper production of butyrate, which was then later on translated into an anti-inflammatory activity, and of course, a strengthening of the gut barrier activity itself. So, in a nutshell, the idea was how to select an in vitro model to perform a study. Well, it all depends on the research question. So first things to look into is about a personalization of the model, which is your target population, which is the age that you want to address. Are we talking about a healthy individuals? Are we talking about potentially diseased individuals? Which is the core area of the gastrointestinal tract that you want to investigate in order to obtain the research question. Second step, customization of the size. Are you in the early phase of development of the product, in which you have, for instance, a lot of lead compounds, you want to look into multiple donors, so you may end up easily into hundreds, 200 arms, hence a need of the throughput, or on the contrary, the need of a system which actually allows you to really dig into the complexity of the gastrointestinal tract and eventually recreate a sort of clinical trial in vitro. And of course, yeah, with all these technologies, you generate a lot of samples. Then, of course, the need of an extensive downstream set of analysis where you look into the bacterial activity, bacterial composition, potential impact at the level of the host, with the idea to, let's say, generate a set of information that allows you to explain what is happening when a given ingredient is ingested in areas of the gastrointestinal tract, which are, of course, a sort of a black box in an in vivo approach. This is, of course, not a replacement of in vivo. In fact, the ideal thing is a complementarity to in vivo. In vivo, you do see what is the health effect of an ingredient. With this type of technology, you can understand what is the potential mechanism of action behind what you see in vivo. Thank you very much for your attention. And of course, if there is any question, happy to answer them.